So we're going to switch gears. This is like a dramatically different talk. So this is really the idea to think about the macrosystem experiment that is, okay, we have all these exciting ideas about how cancer cells could be inhibited, they're in the market. You know, how do we accelerate that process of getting new ideas through the process of drug development to an accepted therapeutic at a time where they matter more? And so what is the problem in that is, you know, it takes 15 plus years to get a single oncology drug to market. It's now estimated to be in the range of over $2 billion to get those drugs to market. We don't have innovation in trial designs. We're not really understanding whether we're hitting our targets. We don't have, you know, any kind of adaptation in our learning. And yet we understand that cancer has many diseases. The old model of having a blockbuster drug which could sustain a $2 billion investment is gone, probably, till the immune system drugs have come forward. Now people are back on thinking that they're going to have a blockbuster drug, but still, it's still unlikely to work the way we thought. And part of the problem is that there's now diminishing interest in drug development. So at the end of the day, who's the big loser? And that's our patients, scientists. Patients first, physicians, scientists, people who are trying to see how they can move these new ideas forward, and the money to test new ideas just isn't there. So this is an unsustainable path. So what can we do to develop a system, a macro system that we can layer on to the field to make things go forward? So this is what we really developed the ISPY 2 trial for. And I would actually say that every stakeholder plays a role in changing this process. It's the drug company. A lot of people like to blame the FDA. They are a drop in the bucket. They are not the problem. I'll tell you, it is the way in which we write protocols, the way in which we think about it, the way everyone wants to have, you know, everything we've done around biomarkers. You know, it is people not willing to be part of trials, not, you know, it's just on every single level, everyone carries the burden of blame and the opportunity to step forward to fix it. So I'll show you some of the ideas that, or what we've tried to do, and just maybe some thoughts, since this is a meeting on neurodegenerative disease, and I'll talk about this in the next panel, but still just some thoughts of, is this applicable to things other than cancer? And I use breast cancer as a model. But it really is a model. So I really try and think about, when you're trying to put a clinical trial together and you're trying to test something, at the end of the most important starting point is patient. Who has the disease? What's the natural history of that disease? When is the right time to intervene in that disease? When can you measure something? What's the most efficient way that you can think about that? And to try and think about what your clinical processes are for how you work someone up, but to integrate that with research. One of my incredible frustrations in medicine is that we practice, but we never get feedback. So in my book, that's not practice, that's just doing stuff. And that we really should merge the systems. I mean, really, after 25 years doing the same thing, that doesn't help you do anything better. So we should merge the process of care and research, and we should have a trial process that mirrors our clinical process that allows what we call adaptive design, is really learn as you go. And that's really, so we should start with trials, but then we should change our clinical processes. And around that means that we have to change our operational infrastructure, our ability to collect real-time data, and our ability to give feedback. And these are all the tools really that you had to put in place. And then you can start thinking about, okay, I'm going to link my phases of trial design. I have to think about, okay, first I have to get safety data, then I have to get phase 2 data, and then it has to be then confirmed in a phase 3 trial. So really, if you're thinking about it, you have to think about the whole process from beginning to end. And the pieces that you can put in place is you can say, okay, it's in everyone's best interest to have a pre-competitive consortium where people collaborate earlier. I think we've sort of put the competitive burden, we've driven that down, which I think has, enormously increased our inefficiency, and really get the regulators on board with this whole concept of master trials and master protocols, which, by the way, they are. So they, again, are not the problem. So I'll give you an example in breast cancer. That's what ISPY-2 trial, ISPY, there was an ISPY-1. There is now an ISPY-3. But there is, that ISPY-2 is really the thought of saying, okay, in breast cancer, who's really at risk to die and to die early? These are people with more locally advanced disease or stage 2, 3 kinds of breast cancers. And it turns out, I'm a surgeon. That's one of the hats that I wear, but it happens to be one piece of my training. But I know enough to know that someone who's at risk to die of metastatic disease, I am not going to fix them by taking their tumor away. In fact, I get in the way because I'm taking the biomarker. You know, everyone wants, what's the optimal biomarker? Well, biomarker is the tumor. So why take it away? So if you can give chemotherapy, if you know you've got someone who, if you don't give them systemic therapy, they're going to die of their disease. And for sure you're going to give it first. Use the tumor as a biomarker to find out whether it works. And it turns out in breast cancer, the order of disease doesn't matter. And these aren't the people, by the way, you know, it was this concept somehow, if we could just screen everybody and find all these tumors early, that we would prevent this. Well, that hasn't happened because this is a different disease. And we're actually increasing the burden of disease by finding stuff that doesn't matter, but we're not stopping this. So these are the people who have high risk, high grade disease where they're likely to progress in the first 3 to 5 years and die. And the burden of this type of disease is often young women. So this really matters. Now, you know, so that neoadjuvant when we say neoadjuvant, okay, that means giving it first. So adjuvant therapy is when you give chemotherapy or whatever it is. So instead of giving it after you take the tumor out, you give it first. It also helps you, if it works, the tumors go away, so you can do much less surgery or much more cosmetic surgery. You can understand the response to therapy, and it turns out that having a complete response is incredibly predictive of a good outcome. So all kinds of benefits from here. And in fact, if they're not doing well, you have at least one other opportunity to try and get it right before they get metastatic disease and die. And this really was our opportunity to say, well, let's accelerate drug development here, and let's figure out, instead of having this be, you start your disease in the metastatic setting and you run a phase 2 and then a phase 3 trial, and that can take about 6 or 7 years, and then you start and give it in the adjuvant setting where you take the tumor out, and then you have to take 5 years to recruit because you're not sure who's responding and who's not, and then you have to wait 5 years to get your results. There's just no way to get the knowledge turn down below 15 years. So this in particular is also a relevant tool to sort out optimal treatments in the molecular era. So we've got emerging biomarkers and tests and therapeutics, and how can you put all this together and have an efficient system? So the concept is to say, okay, let's think about changing the way we test promising drugs where they matter most. So bringing these early. So as soon as you have safety data, don't go to the metastatic setting. Go right to the primary disease presentation, where if you actually make a difference, you're going to save that person's life. And so far, we haven't cured anyone with metastatic disease, and I'll tell you why I think it's so important there too. Is, you know, we just may be skipping over drugs that really matter. You can use biomarker and imaging guidance. We can use adaptive design. I'll show you how that looks. In the IT solutions, obviously, if you're going to do real-time data collection, you have to have things that work for that, and leveraging this whole concept of pre-competitive collaboration. So here's the poster child, and the reason why I would say that I think we're testing drugs in the wrong space, and I think this is true also for the neurodegenerative diseases or some of the diseases of aging, but it's particularly problematic because how early do you start? So this is CML, and Gleevec is the drug that has revolutionized the treatment of this. On the accelerator, so here we see what's happened over time. So these are decades. So the bottom line are the 1975 to 82 and 83. This is before Gleevec. Then you start to see the introduction of Gleevec in the 90s, and now we sort of have Gleevec and all its derivatives. And you can see what's happened to survival over time. It's gone way up. Now, if you sort of look at treatment of people who present in the accelerated phase, that's sort of my equivalent of metastatic disease, on the left, we really haven't moved the needle. So I think it's important not to wait too late in the disease process, even if it's challenging. I mean, we have been called unethical and all kinds of things, you know, any number of bad names because we're trying to push the testing of things early on. But I think the test is, will patients want to be on the trial and will they accrue to this? And we have a lot of advocates involved in it. And, you know, my point is that you know, the universal lesson may be that picking an earlier endpoint for intervention may be really critical, especially if you don't want to miss the signal. So our goal was to screen a series of novel agents in combination with standard chemotherapy in the neoadjuvant setting. So again, what is our clinical process? There actually is a standard backbone. It doesn't have to be perfect. You just have to pick something that most people would agree, here's your standard therapy process. Now, because we have we have drugs that work, they're not great, but they work. So you can't, there's no way you can start a trial where you don't offer that. So so pick a backbone and then add to it. That's the idea. So we said, okay, our endpoint is going to be an early endpoint. We're going to look at complete resolution of tumor. The tumor goes away. That's our endpoint. We call that pathologic complete response. And we set a very high bar because we're screening for drugs with a big impact. We're not looking for the little, you know, part of our problem is that if you will study if you want to make sure you don't miss anything, how are you going to get your pipeline through? How are you going to find the drugs that have the really big signal? So that's kind of tough too, and you have to talk drugs into it, and people are afraid to give you their drugs if you're going to let them fail. So the idea is that you would have an 85% predicted probability of success in a subsequent phase 3 trial. And we're trying to get away from these 10,000 adjuvant person trials, right? So then we want to accelerate the process of identifying drugs that are effective for specific breast cancer subtypes. We know now from all biology that breast cancer is many diseases, right? At least 8 or whatever it is, so we want to do that. With the idea that we're going to reduce the time and cost numbers of patients to get these drugs to market. So this is the overall trial design. We have a 2-part consent. I'm going to go into that a little bit because we use biomarkers to try and enhance our pool of patients so that we're really getting people who have the right biology to be testing chemotherapy plus new agents. So we have people who have early risk, high risk for early recurrence. And that's also something you can think about in diseases of aging as well. And then at the second, once we sort of make sure they have all the things in place, we can then consent them and they go either to the standard therapy or they get chemotherapy plus some novel agent. We monitor them with imaging, and at the end of the day, we look at pathology along the way and at the end, because we want to find out, did you hit the target? If the drug, a lot of times drugs come on the market and the trial doesn't work and they have no idea if the idea of the drug was bad or that it just didn't hit its target. And that's a discipline that's lacking. So you got to figure out how to do that, but you have to be efficient. So one of the things that we do is we say, look, well, we want to make sure that we're using an FDA-approved test. This is an FDA trial. We did this in collaboration with the FDA, so we used something that's on the market that's reproducible and said, well, if you really basically have a low proliferative lesions in a standard way, you're out. If it's ER positive and mammoprint low, you're out of we're not going to randomize you. But we've now taken that collection of patients and we're starting to develop drugs for that group, because that turns out to be a high-risk group of patients still, and they're not responding to hormone therapy and they're not responding to chemotherapy. That's an opportunity to try something different. But at the end of the day, we have to come up, but path-CR doesn't matter, so we have to come up with a better response. And some of that's actually looking at the stromal enhancement and the stromal background and how that changes, which is sort of interesting. Anyway. So you may hear a lot of things, the FDA, Janet Woodcock in particular, likes to talk about the master trial protocol or master IND. What does it mean to have that? That means that you have a trial and you can look at multiple drugs, but you don't write a new trial every time you want to test a new drug. And so what you do, the main body of your trial contains the details of the trial, but doesn't have the investigational agents. Your summary is in another appendix, and each drug comes in as a drug-specific appendix, and it's a simple thing. We're actually down to being able to get approval now in 3 or 4 weeks, and now we're going to do a central IRB process, and we may be able to do it in a week as we want to add a new drug in, and that's enormously efficient compared to what it often can be 2 years every time you change drugs to try something different. And so the idea is over time, a patient comes in the trial and we watch and see what happens to the MRI. That informs our randomization engine. We get the pathology back, and that further updates the engine. And the next person coming into the trial may get a different drug because of the experience. Someone who has an ER-negative tumor, it's not responding to drug B, but better responding to drug C, may be more likely to get to drug C, that next patient. So that's good for patients. It actually turns out to be not so great for the biomarker analysis, which we didn't anticipate as well, but I'll talk about that in a minute. But so that may work that over time a drug may drop, another one can seamlessly be put in, and the same thing if it's graduated, seamlessly put in. So for us, we know a fair amount about biology, so you start with your best stratification, your best biomarker. So that's kind of the basic concept. So for us, we have these established biomarkers, we have our qualifying biomarkers and the exploratory biomarkers. So, people said, oh, well, we want to test the new biomarkers. Well, if the new biomarkers aren't proven, you really can't disrupt your whole trial and say, okay, that's the way it works. And Don Barry, who has done the adaptive design, often says, we always have these ideas and sometimes we're really surprised. So let the trial learn, let the trial tell you. But that doesn't mean you can't take what you think is, if you've got an EGFR mutation, you can't take, phospho-EGFR, or we would have, whatever, a cell line predictor, whatever you think. That is then a qualifying biomarker, has to be done in a CLIA lab, and we're done, a lot of those. But, many of the ones we thought would work don't work. This is not the definitive trial to test that, and we understand that, but we can still look. So if it's got a big signal, and we have some that actually are, so that's good. And then we have exploration. I think that will help as we accumulate why are certain people just not responding to these backbones. So I think that's good. And here's our, we use the midpoint of the high membrane, so it's either moderately high or very high. And that's often a measure of proliferation, looking at hormone receptor positivity or negativity in HER2. So we actually have identified 10 total signatures, and those are the signatures that we evaluate in the trial. And we begin the trial with each time a drug comes in, it has equal randomization probabilities, and every time you add a drug, it can ask, did you meet the graduation futility, did you meet futility or graduation? If not, you continue the trial for that drug and you keep revising until it drops out of the trial. So what does that mean when you're presenting things? That's a different, we're not saying here's a number, we're saying we're not telling you exactly what that number is, because it's not a straight randomization either, but it's a distribution, it's a probability distribution. And so you say, well, here's your control and here's your agent, and the midpoint of this is the estimated probability of a complete path response. And that's really, and the shape of that curve and the distribution gives you a sense, how robust is it, and can I take it forward? And am I going to take it forward to that phase 3 trial? And this is sort of an example of one of the drugs that we did graduate, neratinib, and showing in our control where our predicted probability was 73% in the HER2, all HER2 positive signature, but 79. It actually had graduated, was 85% when we first stopped the trial, but then it dropped a little bit to 79%. But it actually persisted by the time we get the PathCR data in. And here is a signature in the hormone positive. It's okay, not so great. So this is going to get tested in the hormone negative. HER2 positive is going to go on to ISPY 3. This just gives you a sense of all the biomarkers that we had that were both qualifying. The black are the qualifying and the blue are the exploratory. And just to show you how if we get a core biopsy, what we've been able to get out of a single core, and this is sort of how we sort of map our cores. You have to be incredibly efficient. So this means everyone has to collaborate. It's not like, okay, this lab gets one core, and another lab gets the other, and it comes to one place. We try and be as efficient as we can, and we distribute it. One person, it's kind of trust, right? Okay, 3 people may want to do RNA screens, pick one place, just do it. And everyone relies on the data, but everyone gets the data back. So it's a kind of different way of working together. Again, more of team science approach, and these things are really important if you want to get these things done. But then this creates an enormous, and then all the data becomes, the tissue isn't the commodity, the data that you generate is the commodity. And part of the problem is that it's hard to evaluate these new ideas and new targets efficiently. Now, we've tried to reduce the complexity to 10 types, but that's still a lot. We've got a lot of biomarkers, a lot of types, so that's a problem. So we have started. I have to say this is harder than we thought, and the big problem is one of the great efficiencies of this trial is we have a shared control arm. So, you know, your chance of being in the trial, you get there's a 4 to 1 chance that you'll be randomized to a new drug. That's great for you as a patient because you want, you know, if you want to get something new. It actually turns out to be really bad if you're trying to evaluate new biomarkers because your control groups are small. So, but you know, that's the way the cookie crumbles. So we realize that we're going to get a signal here, but it's not going to be perfect. So we're thinking about it. I think that there's going to be more opportunity once, you know, every arm that graduates, you have to wait till everyone's graduated so you can put it into the database. And so we're starting to get to the point where now we're going to have enough data to really do a better job with all these biomarkers. But anyway, it's, you know, you have to adjust for multiplicity. You have to, you know, think about how you go about really getting and looking for a signal. But remarkably, we actually have a couple of really strong signals, particularly in our DNA repair. We looked at PARP inhibitors, which of course are, you know, supposed to work in people with just BRCA, but it clearly is more than that because, you know, we have all the triple negative cancers responding, and so our DNA repair measures all seem to be very strong predictors, and it's much broader than just having a mutation in BRCA1 or BRCA2. So that's actually pretty interesting. I think one of the things that you have to do, again, if you're trying to do any big effort like this is, you know, the clinicians win, the patients win, the biomarker and the research people win, and they get to test some of their hypotheses. If everyone comes together and says, okay, we're going to work as a team, how do we work as a team? How do we get the stakeholders involved? So you work at it at every point you have to do that. And we did that with our IRBs, this really complicated trial. How are we going to figure out how to get that through the IRBs? And actually, our IRB chair, John Helden, said, you know, get everyone in the room. So at the time, we only had 8 sites. We got all the IRB chairs and their PIs and said, okay, here's the trial. Here's why we want to do it. Tell us what you'd like us to change to make it look better. And when I got to the end of the board, I said, okay, now if you want to add one, I have to take one of these things off because my staff are in the back of the room, are going to kill me. So you know, but people then said, okay, here are the things that I don't like about it. We changed the trial, and you know, we've had now 256 total approvals, amendments, et cetera, and not one has been rejected. And we're down to really fast turnaround. So these things can happen, but you have to educate people, bring people together. It's sort of like respectful. There's like no way you can drop something really different on someone's doorstep. I mean, if we had written this trial and sent it to the FDA, it would have been summarily rejected. But what we said was, okay, here's the problem, sit at the table, help us figure this out. They're part of the design process, so of course they're going to approve it. But you know, so that's how it works. So, and we also have to figure out how to give credit. You have to think about, you know, people work really hard to put people on. Normally people get credit by being the PI of the trial. So the first thing I have to say is, like, I'm not going to get credit. I don't get to be a drug chaperone. I don't get to present because, you know, that's not my goal. My goal is to make the trial work. So you pick, you know, you don't make the trial be the way you give credit. Here, you chaperone the drug, you shepherd it through the process, you respond to all the, the queries about complications, you work on developing the protocol. So we've found ways to involve people in different ways and give them credit for it. And that's actually been a really, and you don't think about it if you're a researcher, you don't think about that. But if you get to the clinic, you got to think about what makes the system work, what makes it tick, because that's what's going to get new drugs to patients and so on. And it'd be nice if that weren't true, but that's the way it works. And if someone's going to put a ton of work in, you have to have a way of rewarding them for doing all that work, because you want them to work on it. You want your project to be their first priority. So you have to figure out, that's important. We actually have 21 sites open. We are screening about 30 patients a month. We have screened now over 1,000 patients. We have over 600 patients who've been randomized and 500 people who've completed trials. We have demonstrated that this model actually works from the macro system experiment. Before this trial, no 2 companies would have put their drugs into the same trial. And they're not competing against each other, they're competing with the shared control arm. that has worked. And we actually now we've just put our 8th drug in, which we hope to open next week. It's an HSP90 inhibitor. And huge number of people working there, over 100 people who work on this trial. And so it's really an important, and from our sponsors, from FDA, we've had to come up with, the Foundation for the NIH was the first, and the Biomarker Consortium was the first way in which we got it going. They didn't really want the ongoing risk. They weren't sure that I was going to be good for finding the money. Of course, I didn't realize that I was going to have to find the money. But anyway, everyone has to step up and do what they need to do. So we found another Quantum Leap Healthcare Collaborative sort of taking on the sponsorship for this. And we're trying to create a common process for how do we do innovation in trials and how do we drive this forward. So that's been, I think, really remarkable. So we've demonstrated that our endpoints work better by subtype. We've got multiple pharma companies into the same trial. We have this infrastructure. We have different ways of distributing credit. We can integrate adaptive randomization, real-time data collection. We have graduated 2 drugs. We have several in the pipeline we should hear more about towards the end of the year. And when we started this, was a pathway people said, oh, this will never work. There's never, we don't want to, and the pharma said, we don't want to work on just something for science. I said, well, what would make it work? And they said, well, if you could use this as accelerated approval, if we could get accelerated approval, that would make the difference. So at the beginning, pharma they would give us the drugs, they wouldn't pay for it because it was just sort of a nice to have, not a have to have. And I said, okay, give me the drug, give me 2 years, and I'll get you accelerated approval. And in fact, we did. We got guidance. The FDA said, well, we want more pre-competitive collaboration. I said, okay, I'll get you pre-competitive collaboration. You help me come up with a path for accelerated approval, which we do, and which we did. And so now we can make this a sustaining model. So I understand that that is really a critical driver here, is trying to think about how can you identify those early endpoints. And so, and we've also figured there's a big delay in trying to qualify drugs, like in the phase 1, so we've started our own phase 1 network to be able to make it kind of go seamlessly. And this is the article that Janet Woodcock and I wrote outlining what the process would be. You take your investigational drug and biomarker, you demonstrate, this is ISPY2, right, that you have a promising drug and candidate-associated biomarker. And it's okay if it's just triple negative or it's HER2. You don't even have to invent a new biomarker. And we may not have a new one to add to it, but that's okay. Then you take that and you show in a confirmatory trial that you replicate the endpoint, then you can get accelerated drug approval and approval of the biomarkers, but you have to continue the drug so that you can show that you get the 3-year disease-free endpoint, and then you can get full drug approval. Now, so part of this is how you implement efficient designs with the use of biomarkers to increase your throughput of your therapeutic agents. The key thing here is having the standing trial network. You got to get your engine running. All these sites, really we've qualified sites to be able to change their approach. At our site, fully 50 to 60% of all patients who present with this are on this trial. And we're trying to get it up to 80%. You've got to design trials that people are going to want to participate in. You've got to get your engine running, get a group of people who are willing to work that way. That's how you're going to achieve that kind of throughput. And that is partly you have to integrate your process of clinical care and research, technologically and culturally. I think that's really important. And then why is the standing trial so important? That's really where a huge number of the gaps are. If every time you start up, you have to build your engine, it takes you 2 or 3 years to get going, you're going to have this big gap. So the idea is if you can have these linked phases of trial development and you accelerate your knowledge turns, you have an agent that enters, you got your phase 1B, someone says, okay, I've got a really interesting molecule. Okay, great. We'll help you run the phase 1B. We'll have it ready, because then as soon as I know the drug is halfway through and it looks like they've got a dose, I can immediately write my protocol amendment. I can have it so that it drops seamlessly into ISPY2. As I'm coming through, I've got that 6-month window where I have to wait before I can announce to the world. We can be working on the biomarkers, figure out what the biomarker is, but then I've got an engine for ISPY3. I'm trying to set up an international engine in 8 or 9 countries where their engines are running. So when something rolls off of ISPY2, I drop it there. That would be a way that we could get this whole process down to 3 or 4 years from 15 years. That would be progress. And then you've got new combinations, and then you can start to think about it, then who's not working. And if it works, you can get your accelerated approval, and if you can confirm that they've got the better signal, then you can get full approval. And if it doesn't work, then you go back to you can just pick anything that's going on. But this is kind of the macro system plan that you have to be thinking about if you want to make this go faster. So these are some things, and I can probably talk about this in the next model. But I think, again, standing trials, standing networks, collaboration, innovation in your trial design, these are really important. Innovation around your endpoints. That means you have to think really hard, whatever disease you're trying to look at, what is a good early endpoint? Where could you measure that sort of in the course of care? How can you make it mirror a process that people will want to use so that people would want to say, well, why would I want to be on this trial? You can't test things if 3% of people are on trials. Diseases of aging, fortunately, unfortunately are common, but that means, you still have to get a process where people are going to participate and make that routine. And we change the culture. We, the community that care about having better options, have to say, well, then I need to be the first to step up to be on a trial. This is good for me, and it's good for the community and everyone who comes by. So that's, again, everyone stepping up. And we are actually really working hard also on the very inefficient processes for how you look at adverse events and things, which is, just makes people say, I don't want to participate in trials because they're a pain in the ass. So, and all that. So anyway, I'll stop with that. Thanks.